Today we're going to talk about the biology of cancer and how researchers at MIT are developing nanosensors for inexpensive tumor profiling. Cancer is a disease of uncontrolled cell division, where cells divide at a faster rate than normal cells and form a tumor. A benign tumor is one that does not spread throughout the body, where a malignant tumor spreads and is associated with many types of cancer.
Here we see an example of a cell going through the cell cycle. The cell cycle is a series of events in which duplication of DNA and cell division occurs to produce two daughter cells. This process is driven by signaling molecules present in the cytoplasm. Many of the relay molecules involved in signal transduction are protein kinases that often act on other protein kinases. A signal cell may have hundreds of different kinds of kinases, each specific for a different substrate protein. Together, they regulate a large portion of proteins that control cell reproduction. Abnormal activity of these kinases cause irregular cell growth and contribute to cancer development. Abnormalities in the signaling pathway are checked in the cell cycle control system. Here, the eukaryotic cell examines internal cues, like molecular signals, and external cues, such as DNA damage, and then decides whether or not to move forward with division. Cancer cells are the rule breakers and do not pay attention to normal cues. They divide excessively and invade other tissues.
Now let's talk about these checkpoints. The major checkpoints are G1, G2, and the M checkpoint. At G1, the cell must choose whether or not to divide. Size, nutrient reserves, positive molecular signals, and DNA integrity are the factors checked here. If all goes well, the cell may enter the synthesis phase and replicate its DNA. At the G2 checkpoint, the cell checks for complete copies of DNA and for undamaged DNA. If errors are detected, the cell will pause and allow for repairs. If damage is irreparable, then the cell may undergo apoptosis, or programmed cell death. The cell suicide mechanism ensures that damaged DNA is not passed on to the daughter cells and is important in preventing cancer. The M checkpoint is also known as the spindle checkpoint. At the end of metaphase, the centrosomes are at opposite ends of the cells, chromosomes are at the equator, and spindle microtubules are attached to kinetochores or non-kinetochore microtubules. At the M checkpoint, the cell searches for straggler chromosomes that remain unattached to the spindle. If a chromosome has been misplaced, the cell will refrain from starting anaphase and will allow time for the spindle to capture the stray chromosome.
Recently, researchers at MIT have designed nanosensors that can profile tumors and may provide insight into how they will respond to certain treatments. This system is based on levels of enzymes called proteases, which cancer cells use to remodel their surroundings. In 2014, Sanjita Bhatia and colleagues described using nanoparticles that interact with a type of protease known as matrix metalloproteinases to diagnose cancer. In that study, the researchers delivered nanoparticles carrying peptides, or short protein fragments, designed to be cleaved by the MMPs. If MMPs were present, hundreds of cleaved peptides would be excreted in the urine, where they would be detected with a simple paper test similar to a pregnancy test. Most recently, researchers are trying to adapt the sensors so they could report on the traits of tumors in a known location. To achieve this, they needed to confirm that the sensors were unaffected by background signals and that might be produced in the bloodstream. First, ultraviolet light activated sensors were used, and it was found that the UV light did not penetrate the skin deep enough. The solution was to cover the sensors with material that is activated by a magnetic field. Upon exposure to the heat of the field, the sensor is released into the tumor. Currently, the nanosensors are not adapted for humans, but once they are, the sensors can be used to determine the aggressiveness of a tumor and aid doctors around the world in the best treatment options.